Okay then, let's start today's session. I'm audible to everyone, right? Okay. Share my screen now. The screen is visible to everyone. Okay. Let's start. And if someone is waiting to get into the call, just let me know. Okay. Okay. So today's session will again have the part two of the cloning or recombinant DNA technology. Part two basically deals with, again there will be like what all types of vectors are there, how you can you know uh, create or restriction digest using endonucleases and all these things will be there, types of restriction enzymes and all. This session will cover those and we'll look at like few questions at the end. Okay. So basically, genetically modified organisms can range from bacteria, plants, animals, viruses, where the genetic makeup of the particular organism in question or in consideration has its genetic makeup modified. Okay. So this was basically the overall workflow of how a genetically modified organism is prepared, and the recombinant plasmids are put into the organism of our interest, and then we get the desired phenotype or genotype. Okay. So basically, what are broadly, if we look at the different enzymes that are used in molecular cloning, the restriction enzymes are there, which cuts DNA at a specific site. It can be non-specific also in few cases, but mostly it is specific. And there's polymerase enzyme, which helps in PCR amplification, basically in adding nucleotides and replicating the DNA, making more number of copies of the DNA molecule. Alkaline phosphatase is used. Phosphatase, as the name suggests, it removes the phosphate group from uh, like reactant or moiety or DNA sequence also. Removal of terminal phosphate group, okay? And then DNA ligase, which joins two DNA strands basically by creating the phosphodiester linkage, okay? Helps in creating the phosphodiester linkage between two nucleotides, okay? Yeah. So, the first is how does this name come, like restriction methylase? What basically are restriction enzymes, okay? So this is actually restriction methylase system and or restriction modification system. This is actually naturally found in bacteria as as a sort of or as a mechanism to protect themselves against viral attack or viral pathogens. Okay, so what happens here is, in general, suppose if we are talking about eco-argon restriction enzyme, it has its site, like this is the recognition site GAA-TTC, and it cuts between G, first G and the second A. Okay, these residues, it cuts in between these residues on both the strands. Okay, now what happens here is, see, when there is a viral attack, the particular genome can be integrated into the bacterial genome so that bacteria can have a memory of that particular attack. All right. But then what happens is, because now if something similar attacks later, then what will happen is bacteria can recognize that particular viral genetic or genome that has entered, and then it can directly chop off that thing so that later invasion won't occur. But then if such a thing is integrated into its own genome, then what will happen is, uh, it will basically, these enzymes, these all these restriction enzymes can chop the bacteria's like bacterial genome, like the bacterial genome, which can again might be harmful for the bacterial organism itself, bacterial cell itself. So what happens is, the usually the parent strand or in general the bacteria, in the bacterial cell, the cell's own genome or the own DNA sequence is methylated. Okay, and methylated this methylation or addition of methyl residues, CH3 residues, onto the nucleotides is done by methylase enzyme. Okay, so what happens is if the uh, particular residue, even in the recognition site of a specific recognition restriction enzyme, is methylated, then what happens is the restriction enzyme won't be able to really recognize its uh, like cut site, okay, specific site to really recognize and cut the DNA sequences. So this is what has been shown here. This is the unmethylated DNA. EcoR1's recognition site is this 6 nucleotide bases, and then it will ideally cut here, forming these sticky ends, and then these fragmented DNA will be later degraded totally. Okay. Now what happens here is methyl adds CH3 group at this third adenine, okay, adenine residue. And then what happens is, if this has been methylated, then the eco-organ restriction enzyme won't be able to really cut or recognize this specific site for its restriction activity. So methylated DNA will not be cleaved by eco-R1 and there won't be any DNA degradation. This is the case in case when suppose the viral genome has been integrated into the bacterial genome as a memory. All right. So in that way, this happens. So this is the restriction methylase system. Now, just few basic things like what are actually palindrome. When palindrome basically is the sequence of nucleotides which reads the same from 5 prime to 3 prime direction of complementary strands. Okay, see here T T A G C A C, the same thing. When we suppose this is 5 prime to 3 prime and this is 3 prime to 5 prime, but from the 5 prime direction, what is the sequence? It's the same as that is being read from the 5 prime to 3 prime direction on its complementary strand. Okay. So this is what constitutes a palindrome. And there are mirror repeats also, which is on the same strand. Here, in case of palindrome, complementary, both the strands are involved. In case of mirror repeats, only one strand is involved, wherein TTAGC, then suppose if you put a mirror, it will read the same on the other side. Okay. So this is what is known as mirror repeat and palindromes. Okay. Just for basic information. Now, what happens is restriction enzymes can create or act at specific sites. So it can create different types of ends. Okay. So sticky ends are the ones which creates basically overhangs. So these are not blunt-ended DNA. These don't create blunt-ended nucleotide or DNA sequences. So what happens is suppose in case of sticky ends also there are various types. What happens here is this is the double-stranded DNA, linear DNA with few nucleotides protruding. The lengths of the two strands can be same or need not be the same. Okay, but see some of this green part here, this 3 prime at the 3 prime end, some of the nucleotides are protruding. Okay, or at this part also some might be protruding. Okay, on this strand as well as on this strand. So there is either we can call 5 prime recessed ended. What happens here is or 3 prime cohesive ended or 3 prime protruding ended. What happens here is 5. This might have been a whole, this is the whole DNA, like original sequence might have been the original sequence. And what happens here is in both the cases, see, so 3 prime end is protruding here. So in such cases, what happens is this is also called as 3 prime protruding ended or 5 prime recessed ended. Recess means there's a gap, right? Recess means a break. Break as in not a nick kind of thing, just a gap kind of thing. So see at both the 5 prime end, there is this gap here, right? So this type of cut or this type of fragments when the DNA sequence is treated with certain types of restriction enzymes, these kind of fragments where the 3 prime end is protruding are called 3 prime protruding end or 5 prime recessed ended. Okay. Or it can be the other way around, wherein the 5 prime ends are protruding on both the strands and 3 prime end is recessed or has a gap here, right? This is what is exactly it just how it looks. Uh, Based on that, the name has been given. Okay. Then there are blunt-ended DNA also, wherein there won't be any overhang created, like it cuts at exactly, suppose eco R1 in the previous case was suppose something cuts here, exactly here, okay, at both these places. So what will happen is, but this is not the case. It should be proper, there should be the two palindromic sequences. But then suppose it is cutting exactly here, then what will happen is this is a blunt-ended DNA. And suppose something is cutting here, suppose between T and A, and here between T and A, then this will produce overhangs or sticky ends. In case of blunt-ended double-stranded linear DNA, lengths of the two strands are same and completely base paired. Okay, so there here these particular overhangs are not base paired. Okay, mixed double-strand linear DNA. So what happens here is absence of phosphodiester bond in any one strand. See here in this case, there, like there's a 3' OH present here, there's a nick present. So 5' to 3' OH and then 5' phosphate here and 3' OH here. There can be multiple nicks in the same or complementary strand, but not contiguously. Okay. Then there might be a gap double-stranded linear DNA. So when we call it as nick, it means at only one of the strands there's a like there's breakage in the phosphodiester bond. Okay, or there's no phosphodiester bond present. When we are talking about a gap, a total gap, then there are multiple such nucleotides absent. Okay, multiple nucleotides are absent from the sequence. So multiple phosphodiester bond is not there, thereby creating a gap, which is broader and multiple nucleotides are absent. And here, nucleotide might not be absent. Only there's a like the bond is not present between, like only one phosphodiester bond is absent here. Here, multiple phosphodiester bonds are not there and nucleotides are actually missing. Okay, this is called a gap and this is a nick. Okay. Now, what happens here is, I think this is again taken from the lecture video only. So restriction enzymes are categorized based on their structure and function. Mostly there are different types of restriction enzymes, and mostly the type 1 restriction system are categorized based on the structure and function. So it can be R type, S type, and M type. Okay. The R, S, and M basically refers to the components of the restriction modification system. What happens here is, see, suppose these are the restriction sites, and R, S, and M are the components of a restriction enzyme. Okay. Now what happens? R stands for restriction here. Okay. S stands for specificity here, and M stands for methylation or modification. Modification refers to addition of methyl groups, like that. Okay. It can either stand for primarily modification, but what happens basically or generally is methylation. Okay. So what happens here is the R component. This component is responsible for cleavage of DNA at specific sites. S is for specificity, which determines the recognition sequence for the restriction enzyme. And M is for modification, which adds the methyl groups. So methyl groups carries out the methylase activity to host to the like, adding methyl groups to the host DNA to protect it from being chopped off by the restriction enzyme. So what usually happens is, first, suppose this restriction enzyme is binding. So based on the S type, it will search for the recognition sequence, the specific sequence to really look for the specific recognition sites. And then once that site is recognized, there will be a closed complex of this particular restriction enzyme. And then it can actually, the R type then come into play, wherein it can chop off the DNA, or suppose either it can chop off the DNA, or if there's a modification that has to be made, then the M type comes into play, which methylates the particular uh, residue in the DNA sequence. So see, this is the same thing. First is recognition of cleavage site, definitely. This is type 2. This is again taken from the lecture videos. So primarily it is type 1 enzymes. Okay. So type 1 restriction enzymes have specific recognition DNA sequences to rapidly identify the site. The enzyme can scan the stretch of DNA through a non-specific interaction and diffusion along the length of DNA. This is what is shown here. Okay, suppose there are multiple restriction sites present here, and this particular enzyme complex has come, so it will first scan. It won't really bind. First is it will scan for the specific sequence of nucleotides and then bind at the specific site. Okay. So while scanning, because it has to scan along the length, so there will be non-specific binding. But once it gets its or reaches the reaches its exact recognition site or recognition sequence, then there will be specific binding. It will bind tightly at that place. Okay. Yeah. To expedite the recognition process, enzyme does not make interaction with the bases. Instead of it, it makes contact with the DNA backbone. Okay. Now binding to the recognition site. Once the recognition site is located, enzymes make specific interaction with the nucleotides present at the recognition site via entry through major pores. All right. Hydrogen bonding, van der Waals interaction play a crucial role in this step. Now, even in case of non-specific interaction or in case of specific interaction, the forces that are acting or the interactions that are acting between the enzyme and the bases are broadly non-covalent interactions. Okay. So here, even when the specific recognition is, the specific sequence is recognized, then also these non-covalent interactions like hydrogen bonding, van der Waals interaction, these are the major ones that help the enzyme to really interact with the specific nucleotide sequence. Enzyme DNA closed complex formation induces major conformational changes in the restriction enzyme. Now, due to such conformational changes, when the DNA, being the substrate of the enzyme, binds and induces such different kinds of changes in its structure, what happens is the closed complex can activate the cleavage activity. This will be CLEA, cleavage activity of the enzyme, resulting into introducing DNA breaks on both the strands to give a fragment with 3' OH and a 5' phosphate group. Okay. Now, what happens here is distinction between self versus foreign DNA. So that was what I was talking about in the first part of the restriction methylase system. So although the precise mechanism of distinction is not known, but based on the available literature, what happens is in the absence of methylation, a closed complex is formed and it allows the proper activation of cleavage activity of the enzyme. This is what is being told as cleavage of closed complex, which activates the cleavage of the DNA sequence. Okay. And but in the presence of methyl group, even hemimethylation. Hemimethylation means not the same, like the residues on both the strands are not uh, methylated. Only some residues on one strand is methylated. The other complementary strand, the residues on the complementary strand aren't methylated. Okay. Presence of methyl group or even hemimethylation on nucleotide does not allow the formation of closed complex, and consequently the enzyme falls off from the DNA because it can't really, uh, because the DNA being the substrate for the enzyme in this case, in this scenario, the DNA won't be able to bind because the unmethylated bases can really fit into its pockets, okay, or the active sites of the enzyme, but methylated ones don't like methylated residues won't really have that particular exact configuration or conformation to fit into the pockets of the enzyme. Thereby, if the enzymatic activity can only be activated when the conformational changes are induced into the pockets of the enzyme. I mean, in conformational changes are induced in the whole structure or into that at the active site of the enzyme, then only the catalysis can take place. But if methyl groups are present, this catalytic activity won't be there. Okay. Now let's come to, I think many of you will be knowing this, how to name the restriction enzymes. So suppose if we are talking about EcoR1 name, so E comes from the genus of this species we are talking about. Suppose in this case it is Escherichia. CO comes from, and it is always in caps, and then CO comes, which is coli or which refers to specific species. With this R or RY13 strain, which refers to the strain of the particular organism we are talking about, the bacteria we are talking about the context. And then one refers to the order of the identification in the bacteria. Then there are numerous restriction enzymes. Suppose if we are talking about HIND2, HIND 2, suppose, or 3 or 1. So there are multiple other ones as well in the same series. So it comes from Haemophilus H and Influenzae species IN. Okay. So this is how it comes, and the strain and then the order. The number refers to the order. Now, there can be different types of restriction enzymes based on their structure, the recognition sites, the sites where they are actually cutting. They can recognize a whole particular length of bases, but they can cut at some other site altogether, or at a site like for in case of EcoR1, GAA, right? So, but it can also recognize certain. There are enzymes it can recognize certain bases or certain nucleotide sequence, but it can cut at some other site altogether. Okay. Also, these restriction enzymes can vary depending on the type of the cofactors required for their function. So there's type 1, which cleaves at sites quite far away from a restriction site, from their actual cutting site. Okay. Uh, and it requires both ATP and SAM. SAM is here is S-adenosyl L-methionine, SAM. Type 1 is a multifunctional protein. It contains restriction division, the R activity, as well as the methylase activities or the M activity that we were talking about in the previous slides. It has been identified in two different strains of E. coli, K12 and D. So, uh, suppose we saw EcoR1 R strain, K12 strain, K16 strain. There are multiple strains of E. coli. Okay. And basically, this type 1 has three subunits, HSDR, the same thing, R, S, and M, restriction showing restriction digestion activity. M shows methyl transferase activity, which actually functions as a methylase, and S is the DNA binding domain, which actually renders specificity to the enzyme to bind at its specific recognition sequence. Okay. And it requires magnesium ions to for its function, and there is non-specific cleavage of around 4,000 to 7,000 base pairs away from the recognition site. I think in the lecture videos, it tells that it's around 1,000 bases. So in the range of thousands, it can really the recognition sequence is suppose GTTCCC, but it is cutting altogether at some other sequence which is around 1,000 base pairs or thousands of base pairs away, maybe in between C and G, somewhere away. But exactly because what might be the reason for this is because there are these three subunits, right? So once it is recognized, it will bind at some point using this subunit, and the other subunit uh, can actually reach that part, or the DNA can loop and come into the vicinity of this particular subunit, the R subunit, so that the restriction activity can take place or digestion can take place of that particular DNA sequence in question. Okay. Type 2 restriction enzymes. This is actually the most commonly used ones in our day-to-day laboratory purposes or research purposes. So the recognition site of this type 2 enzyme is around 4 to 8 nucleotide long, and it cuts the DNA within the specific site, like EcoR1 that we saw. And due to this feature, type 2 restriction enzymes have applications in genetic engineering for cloning purposes. This is also composed of three subunits: M, modification, or responsible for modifying or methylating the nucleotides or DNA residues. R is the restriction activity, and S for rendering the specificity. Type 2 enzymes are of diversified nature and are further classified due to the unique feature of each class. Okay. So now type 2, type 1 over, now like within the type 2 restriction enzymes, there are multiple types of type 2 restriction enzymes again. Type 2B, these are multimers. These cleave DNA on both sides of the recognition to cut out the recognition site. Okay, like these are the examples: BCG1, BPL1. And then type 2E, these enzymes cleave DNA after interacting with two copies of the recognition sequence. After recognizing, suppose a four base pair sequence they recognized, this was the first target for cleavage, and then the second same sequence has to be present, so that they will be cutting at the first recognition full base pair sequence, but the second has to be present. so that this particular cleavage activity can take place. Okay. Example of such type of enzyme is NAE1. Okay. Then there's type 2F, which can interact with two copies of the recognition sequence, and it can cleave both the sequences at the same time. Okay. So suppose in case of in general, when we are talking about any restriction enzyme that is cutting at a specific sequence. So basically, how many fragments will be if it's a linear DNA, if the if a particular restriction enzyme is cutting at only a specific site, suppose we are talking about type 2B. Okay, then how many copies of or how many fragments of the DNA will be formed? Can anyone tell? So I was asking, suppose here in this slide, I'm talking about particular restriction enzyme is cutting a linear DNA at a specific site. Okay. So how many fragments will be formed in case of linear DNA and in case of a circular plasmid DNA? Yes, yes, good. So now next what I was talking about, why did I ask this question? Because uh, here, see in this case, I will return to the presentation. Here, now, because in cases, the example that I asked about was that the DNA was being cut at one specific sequence. But in these cases, suppose E and F cases, they, in E case also, there is only one cleavage site. But in F cases, they interact with two copies of recognition sequence and cleaves both the sequences at the same time. So then in case of linear DNA, if two cleavages are formed, right? Two sides are being cut. Then in case of linear DNA, there will be three fragments. All right. And in case of a circular DNA, there will be two fragments because two recognition sequences are there and both are being cleaved. Okay. So just a small thing to keep in mind. No need to, I don't think this is important to memorize as such, but just to maybe just visualize the thing, what might be happening. Okay, how can we really use this particular activity for our experimental purposes or for experimental design? Okay. Then there's type 2G, which is a single subunit is enzyme and requires SAM as a cofactor. Type 2M is there, which can recognize and cut methylated DNA, like DPN1 enzyme. Type 2S is a dimeric enzyme having like dimeric subunit. It cleaves DNA at a defined distance from their non-palindromic asymmetric recognition sites. Okay, so this particular 2S recognizes non-palindromic sequence. And there's type 2T with two different subunits which recognize palindromic sequences. Okay, so these are just different varieties of type 2 restriction enzymes we were discussing, just briefly talking about. And then there's type 3 restriction enzymes. The recognition site has two separate non-palindromic sequence arranged inversely oriented. And the cutting site is nearly 20 to 30 base pairs away from the recognition site. And this type 3 enzyme is composed of two subunits, res and mod. The mod subunit is required for modification or methylation of the host DNA, and res subunit is basically for restriction activity. Okay, or cutting the unmethylated DNA strand. Okay. Now, just few things before because we are talking about restriction enzymes here, and while setting up a restriction, I think there were two places where how to really what might be the components of a reaction mixture during setting up a restriction enzyme reaction and as well as ligation reaction. We always have to use enzymes. Okay, so there are certain things that needs to be kept in mind, wherein we talk about units of enzyme to use in a reaction. Okay, so when we talk about the enzyme unit, this is quantifying an enzyme's catalytic activity. So one unit is basically micromoles of the enzyme that is catalyzing in a particular time frame. So one unit is the amount of enzyme that catalyzes the conversion of 1 micromole of substrate in a minute under specified assay conditions. Okay. And these conditions, the assay conditions that we are talking about, it can include temperature, pH, substrate concentration, which yield the maximal substrate conversion rate for that particular enzyme. Because at different temperatures, the activity of a particular restriction enzyme can change. Okay, so ideally what happens in many cases is that suppose EcoR1, and again, there are companies, different companies that supplies the enzymes. So what I'm talking about now is a daily laboratory, like what we do in labs and like during setting up experiments. What we do is the enzymes might come from different companies. Now the composition or the buffer composition, the solvent environment for the enzyme might be a little different. So the incubation time for a particular reaction, the temperature for a particular reaction might vary. So there's a certain type of, suppose for setting eco-carbon reactions from a particular company, then it is the reaction is set, the restriction enzyme digestion is set at around 65 degrees Celsius. Okay, or 37 degrees Celsius. And then what happens is, after a certain point of time, you have to deactivate the enzyme. What so it can be done at 65 degrees Celsius or at 80 degrees Celsius to deactivate the enzyme, because what happens is, if we incubate our DNA sequence for a long time using the particular restriction enzyme, what might happen is it can chop or digest the DNA at non-specific ends. We just need to cut the DNA at a specific site, but if the enzyme remains um, like remains in the vicinity of that particular sequence at its optimum temperature, what might happen is it can introduce non-specific cuts at the DNA, which we don't really want for our experimental purposes. So there's a certain time point when this enzyme has to be used for the digestion reactions, okay, as well as at a particular temperature. Because usually the way to deactivate an enzyme during our day-to-day laboratory experiments, what we do is we increase the temperature uh, at a specified range so that the activity is deactivated. Okay, it is denatured, or it won't be catalyzing. The reaction loses its catalytic activity. Then now I was talking about enzyme activity, which is expressed in units per milliliter. This is the unit, which is the amount of enzyme that is catalyzing the conversion of 1 micromole of substrate in a minute. And enzyme activity is like the concentration, like is the unit of enzyme being used per milliliter of reaction volume. It relates to the concentration and varies depending on the enzyme's role in metabolic pathways, DNA replication, gene expression, and other conditions, okay, or other metabolic pathways. And there is this turnover number, or kcat, which represents the maximum number of substrate molecules that an enzyme's active site can convert to a product per second when fully saturated with the substrate. Okay, these are just the definition that I'm talking about, because sometimes suppose I have been talking about the activity of the enzyme. So what exactly is, how do we define it in the literature? This is what is the definition of an enzymatic activity. This is the definition of what one unit of enzyme refers to. So this is just for your, like you should have this in mind when we talk about such terms. This is actually what it means. Okay. And then star activity. So what I was talking about, there might be, if we suppose incubate the DNA sequence with the restriction enzyme for a longer time, there might be star activity or non-specific activity of the enzyme. So under non-standard reaction conditions, suppose in this case, what I was talking about is the incubation time. That is, if I uh, increase the incubation time as opposed to what has been suggested, what has been standardized. So then some restriction enzymes can cleave or cut the DNA sequences similar to but not identical to their defined recognition sequence. Now, the factors contributing to star activity can include glycerol concentration, because it makes the reaction volume very viscous. So it might not be optimum. The viscosity or the thickness of the medium viscosity of the medium might not be optimum for the enzyme to really function. Then buffer choice, what kind of what are the components of the buffer inside? What is the reaction time that I was talking about in the example previously, and the presence of organic solvents. Okay. So the star activity of an enzyme can lead to cuts at non-canonical recognition sites or like non-specific recognition sites, or even it can also lead to complete loss of specificity. Okay. Now, now let's look at some of the enzymes which, so initially we saw that different kinds of sticky ends that can be, or different kinds of overhangs that can be produced, 5' resistant, 3' resistant, and blunt ends. Okay. Now let us look at the some of the enzymes that creates such type of DNA sequences, overhangs or blunt-ended DNA. Okay. So eco-R1 generates 5' overhang because it is cutting at a non-symmetric part. Okay. GAATTC. Now, if something was cutting in between A and T here, the third A and the fourth T here, then it would have been a blunt-ended DNA sequence. See, this is the overhang we are talking about. Okay. Now, here, this is, can anyone tell what kind of, like, we are talking about 5' overhang. So, uh, this can be, like, the previously we were discussing about recessed ends. Okay. So in this case, when eco R1 is cutting the sequence, what is the type of recessed end that has been generated in this case? Can someone try? Okay, so because here in this case, what is happening is recessed means there's a break or a gap. So this is 3' recessed ends that has been generated. These are 5' protruding ends or 3' recessed ends. On the other case, this is an example of an enzyme PST1 which is generating 3' overhang, and see here, this is 3' overhang or 5' recessed ends. Okay, in this case. Recessed means like there's a gap in between. Okay. Now, these are some of the examples of blunt ends generators, like HAE3 generates blunt ends. See, it is exactly cutting at a very symmetrical site, generating these blunt-ended DNA fragments. There's SMA1 or SMA1. This also cuts at a very symmetrical site between G and C. But XMA1 again produces is a sticky end generator. Okay. Now there are some terms related to the restriction enzymes. One is isochizomer. These are enzymes that recognize the same target DNA sequence and cleave it in the same way. Okay. Suppose SPH1 and BBU1, this recognizes the same sequence, and exactly the restriction site is also the same between this C and G. Okay. This is isochezomers. Then there's neochezomer, which are enzymes that recognizes the same target DNA sequence, but it cuts at different points. Okay, suppose SMA1 we are talking about, it cuts in between C and G, creates blunt-ended DNA sequence, blunt-ended fragments, but XMA1 cuts at recognizes the same sequence 3C and 3G, but it cuts at in between these the first and second C residue, which creates blunt sticky ends. That's what we saw here. SMA1 is recognizing the sequence triple C triple G, but it is cutting at a symmetrical site, thereby creating blunt-ended DNA, whereas XMA1 is cutting at an unsymmetrical site, thereby creating sticky-ended DNA fragments. Okay. So basically, SMA1 and XMA1 are neochizomers. Okay. And there is another term called isocodamers in case of restriction enzymes. These are the restriction enzymes that produce the same nucleotide extensions but have different recognition sites. Okay. Suppose BAMH1 cuts in between these two G residues and recognizes this particular sequence. Okay. And this SOU3, uh, which again recognizes a particular DNA sequence but cuts at, again, if it is cutting before this G what happens is this is also creating GATCC. This is also creating GATC. Okay, extension. So isopodomers are the restriction enzymes that creates the same type of overhangs. Okay, and neosesomers are recognizing the same DNA sequence. The recognition site sequence is same, but the that sites are different. But sites are different. And isosesomers, what happens is they recognize the same DNA sequence, the recognition sequence is same, as well as the recognition sites are same. Okay. Now, what happens in a DNA ligase reaction? We were reading about, okay, DNA ligase joins a nicked DNA strand or something like that. Okay. So what happens here is um, this is an enzyme which requires ATP for its function or NAD plus as a cofactor to catalyze the ligation reaction. Ligase is processing ATP to generate AMP, thereby releasing the two inorganic phosphate. And then AMP is making adduct with enzyme here. Okay, AMP enzyme complex. Okay, here suppose um, there's this DNA strand with 5' phosphate. There's this DNA strand with 3' OH. Okay, free 3' OH and free 5' phosphate group. Now, this particular enzyme, DNA ligase, uses ATP to form AMP enzyme adduct and releases an organic phosphate here. Okay, PPA. Now, this ligase AMP complex that is being talked about, this is the one that is being referred to. Okay, AMP enzyme. Now, this complex is binding to the 3' and 5' end of DNA, bearing nick and bringing them together. Okay, and when those two ends are brought together, AMP is released and phosphodiester linkage is formed between the 3' and 5' end to seal the nick. Now, this particular enzyme adduct, the complex has formed with AMP. Now, this free 5' phosphate DNA containing fragment, and it first brings in this, and then when this particular 3' like the DNA fragment containing the 3' prime OH, like freely having 3' OH, that particular fragment comes near it. And what the enzyme does is AMP is released, and these two, the phosphodiester linkage is formed. Thereby, AMP is released, and these two DNA strands are joined together. Okay, so DNA ligase, what it does is it actually utilizes one ATP molecule to join two DNA fragments. Okay, like to create, or rather to create one phosphodiester linkage. Okay, now when we talk about alkaline phosphatase, what it does is it removes the terminal phosphate groups. Now, suppose this is 5' phosphate, free 5' phosphate. We have digested it using restriction enzyme. Okay, this we are talking about the circular plasmid DNA. This is 5' phosphate, 3' OH, 5' phosphate, and 3' OH. Okay. Now, if we use ligase enzyme, what we saw in the previous slide, like it will use ATP to join the two fragments. Okay. Like this will seal the gap here and introduce phosphodiester linkage. But when if we are using phosphatase, suppose we for some reason we don't really want to, what happens is suppose if these in case of blunt ends, or when we have if we leave the vector as such without introducing any other any other any insert or something, so vector can always ligate to itself, ligate with itself without introducing the insert DNA. Okay. So what might happen is, so what people usually do is, if you remove the like terminal phosphate groups, then there won't be any reaction going on between like 2, 3' OH can't really join together to for a continuous DNA strand or DNA sequence. Okay. So what happens here is if phosphatase is there, see here, like it removes, like in place of 5' phosphate, there's 5' OH now and 5' OH now. Phosphate groups, terminal phosphate groups have been removed by phosphatase enzyme. Now, suppose we are having our DNA of insert. Now, without any, because this particular insert DNA has 5' phosphate on both the strands, all right, like free 5' phosphates, how we saw here. this particular DNA can then ligate to this 3' OH, and 5' phosphate can be acted upon by ligase to join to create a phosphodiester linkage, and here as well in this 5' phosphate and this particular 3' OH can join together to form a continuous strand. Okay, but what happens here is, now see, if there would have been a 5' phosphate here in this case, if this would have come, this would have been a continuous strand, right? But in this case, because phosphatase enzyme was used and the terminal phosphates were removed, we don't have free phosphates here, right? We have 5' OH groups here, and there's 3' OH groups. So 3' OH and 5' OH, there won't be any bond formed. So this is what is, there is a nick. There is an absence of a phosphodiester linkage here. All right. So on both the strands, there will be a nick. Now what happens here is, this circular recombinant molecules with single-stranded nick at each end of the vector. This is what is being talked about. Okay, one nick here where 5' phosphate was removed, and one nick here at the other strand where 5' phosphate was removed. This portion usually transform as efficiently as the fully ligated product. Fully ligated means, suppose in this case scenario when terminal phosphates were present, if this would have come very nicely, this 5' phosphate would have joined here, and this 3' OH with this 5' phosphate would have joined together to form a phosphodiester linkage without any nicks present. Because nick is there because we can't really form any bond or the ester linkage between 5' OH and 3' OH. Okay, but what happens usually here is, when you transform this particular vector with the nicks present on both the strands, what happens is, once it goes inside the cell, the cellular machinery, suppose you're like we read about transformation, right? Like putting in a foreign plasmid or foreign circular plasmid into, suppose, a bacterial cell or E. coli. So what happens is, after transformation, the nicks, these can be repaired because this is getting, replicated. So all these machinery that represent like the natural metabolic pathways that are present inside the bacterial cell can take care of these particular nicks. So the phosphatase treatment of the vector, so this is what is written here. Circular recombinant molecules with single-stranded nick at each end of the vector portion usually transform as efficiently as the fully ligated product, and the nicks are repaired in vivo. In vivo means inside the cell. Okay. After transformation. So phosphatase treatment of the vector molecules, wherein we are removing the terminal phosphate groups, in this case the 5' phosphate groups. So the phosphatase treatment of the vector molecules increases the proportion of hybrid DNA molecules in the population of transformed cells. So what is this doing here is, see, if we have introduced this cut, this vector can, without the DNA also, this vector can fully ligate, okay, with itself, and then this itself, without the insert DNA only, without the genetic sequence of our interest, this whole black circle or black like this black sequence can get into the cells without any, without at all serving our purpose. So if we really remove the terminal phosphate from here, and then the DNA, only the ligation or only the phospho that ester linkage will be formed. Only this, only in the presence of the terminal phosphate groups, which is present in the insert DNA of our interest, right? So what will happen is that DNA will react with the 3' OH group, and thereby forming this particular recombinant vector. Now, this can, this in turn will be getting inside this whole mixture when we transform it, when we put the whole, uh, when we introduce these cells inside the bacterial cell during transformation. What will happen is, we can expect more number of these recombinant molecules to be present rather than the vector ligated to itself. Okay. So this is one of the reasons that phosphatase treatment is given to remove the terminal phosphate groups. Alright. Next. Now, what happens sometimes is, suppose in case when we were talking about the, suppose if this would have been a blunt-ended DNA, but in order to prevent its self-ligation, what also can be done is people can use linker molecules. Suppose this is a plant-ended DNA with 5' terminal phosphate, 3', this is OH, like on either side, this is 3' OH. Now they use certain recognition sequence for restriction enzymes that create sticky ends, okay, or overhangs. So suppose for EcoR1, the sequence is GAA-TTC. All right. Now this particular sequence is being used, and what will happen is now you ligate this particular sequence. This is blunt-ended DNA. Now this blunt-ended DNA has free 5' phosphate group, free 3' OH group. This linker sequence also has free 5' phosphate and free 3' OH groups. All right. Now using ligase enzyme, we basically join these two sequences. Now what is happening here is you have the blunt-ended DNA, that is primarily the DNA of your interest, sequence of your interest, suppose, and or the vector. Suppose we are talking about the vector or the DNA of our interest, whatever. Now what will happen is these recognition sequences for certain restriction enzymes have been ligated at both ends. All right. But and then this the recognition sequence for the specific restriction enzyme that has been ligated, those restriction enzymes are introduced here into the reaction mixture. In this case, it is EcoR1. Now, what EcoR1 will do? This will cut at this particular site between G and A, GATTC. This is its recognition site. Now, this will cut at this particular sequence, and then it will create overhang. Now, when you have this particular overhang, this can again be ligated very specifically with your insert or with your vector, either way, whatever, wherever you are adding this particular linker region, this can be done, thereby creating the sticky end and preventing the self-ligation of the vector molecule and giving us much more specific like recombinant molecule. Okay. So let's go through this paragraph once. Linker molecule, in case when linker, what happens here is an amplified foreign DNA may have restriction enzymes at their terminus to give cohesive end. This is being talked about cohesive ends to facilitate ligation into the vector. But in cases when foreign DNA is a genome product, also, yeah. Suppose if there is blunt-ended DNA, that is one case. Or when the foreign DNA is a genome product and there is least possibility to keep restriction enzymes at the ends. What happens is usually when, I think in the previous few slides, slides, I guess, there have been cases where we are introducing, we were talking about how to generate forward primer, reverse primer, right? So we were introducing the restriction sites and the primers, thereby we were getting for clone, right? So, but in cases, suppose when the foreign DNA that needs to be introduced into the vector is a genome product and there is least possibility to keep the restriction enzymes at the ends, because if restriction enzymes or restriction sites or recognition sequences are present in a genome product, then that genome product will be chopped off, right? And that will like completely degrade the cellular machinery, all right, or completely kill the cell. Okay. So we want that definitely. If we want the whole intact gene, in case of like while introducing this particular foreign DNA for using this foreign DNA for cloning purposes, so we can't really have restriction enzymes at the ends naturally, all right, because it might introduce these things. What happens is cloning of these fragments is facilitated by the help of linker molecules. Linker molecules are the short double-stranded DNA. This is what is being talked about. 8 to 10 nucleotide long and has restriction sites at their ends. Okay, in this case, EcoR1. Now, this example has been talked about like 4x. This is again a name of an example of a linker molecule here. 4x is a typical linker molecule with EcoR1 site as shown in this figure. Okay, this one that is being talked about. And this is the foreign DNA of our interest. And the linker molecules, the 4x linker molecule, can be attached at both ends, having EcoR1 restriction sites. Now, if the linker molecules are attached at both ends of our DNA of interest here, then EcoR1 digestion will create overhangs here. Vector will also be digested with eco-R1 having the same overhangs. Thereby, when the vector and the insert will be ligated using ligase, then it will, like very specifically, like probability of forming the very specific recombinant molecule will increase. Okay. Now, linker molecule is incubated with foreign DNA. Here, this is being talked about foreign DNA and linker molecules, uh, and ligated by the action of T4 DNA ligase to generate chimeric DNA. Okay. And the chimeric DNA is digested. Chimeric means there are two different uh, portions in the same molecule. The chimeric DNA is digested with eco-R1 to generate cohesive ends or sticky ends here in this case. It is now incubated with eco-R1 digested vector in the presence of DNA ligase. This is eco-R1 digested vector. This eco-R1 digested vector and eco-R1 digested linker molecules that were already ligated to the foreign DNA of our interest. These two are then recombined or joined together using DNA ligase to get circular clones. Okay. Now there can be adapter molecules as well, wherein what happens here in this case is, suppose this is your genomic DNA having blunt ends, and adapter molecules can have uh, these, these sticky ended portions having OH groups here. So what happens is again you use these small fragments with DNA ligase. In the presence of ATP, what we were seeing in the ligase reaction, it will use ATP forming AMP and enzyme complex that will join these particular small stretches at both the ends because it has free 3' OH. This has free 5' phosphate. So these will be joined, and then what will happen is, because phosphatase removes phosphate groups and kinase will phosphorylate residues on the sequence of our interest, right? So then what happens is you expose this particular ligated fragment into like to kinase treatment, and what kinase does is it adds phosphate residues at these overhangs, and thereby the vector is also, this is your insert, thereby creating a well, like an insert having the proper overhangs. The vector is also digested having these complementary or like compatible overhangs, thereby ligating these two molecules are then ligated, the vector molecule, this one, and the insert after kinase treatment is ligated to form a clone. Okay. Adapter molecules are 8 to 10 nucleotide long double-stranded molecules. These are the ones being talked about with flanking DNA sequence to provide cohesive ends. These are the cohesive ends, these protruding ones. Okay. These cohesive ends have free hydroxyl group to facilitate efficient ligation to the vector. Chimeric DNA containing adapter molecule is incubated with BAMH1 digested vector or any molecule that we are talking about. In this example, it is BAMH1 or it can be any other restriction enzyme that is actually creating sticky ends, producing overhangs. Incubated with BAMH1 digested vector in the presence of DNA ligase to get a circular clone, this final product that we are talking about. Okay. Now, what are the criteria for good vectors? It should definitely be of low molecular weight. The low molecular weight or size actually gives a number of advantages. How? The small size vector is robust towards shear stress and easier to handle. This is what was, if you remember from the previous class during isolation of plasmids. This is because the plasmids are small, that alkaline lysis and denaturation was actually playing a role in separating the plasmid DNA and the genomic DNA, because genomic DNA is comparatively like more huge as compared to the plasmid DNA of interest. Okay. As well as it's easier to handle. In addition, after ligating foreign DNA into the vector, the size of the resulting recombinant DNA will be small and it will be easier to deliver the recombinant DNA into the host cell. Alright. Now, post entry into the host should give phenotypic changes. Another important feature is that the vector DNA should give additional phenotypic changes in the host cell so that the recognition of transformed cells will be easier. Basically, what is being talked about here is a selective stress, like what we were talking about, the antibiotic resistance. Suppose a particular naturally occurring, suppose a particular cell A doesn't have resistance to ampicillin. But then when particular plasmid with a certain sequence has been introduced into that cell A, what happened is that plasmid was having ampicillin resistance gene. So if the plasmid is actually has been taken up by the cell A, then only those cells can be thrown onto an ampicillin medium, like a medium containing ampicillin. Whereas the ones which did not take up the plasmid would be able to grow on ampicillin containing medium, because in general the cells, the type A cells that I am talking about, this is just for an example, that won't be having naturally the ampicillin resistance characteristic. And when the plasmid having ampicillin resistance is introduced into the cells, then only the ones that have nicely taken up the plasmids, those will be able to grow on ampicillin, like a medium containing ampicillin. Okay. So this is what has been referred to. Suppose the ampicillin resistance that I am talking about here in this example refers to the phenotypic changes that have been mentioned here. Okay. Now multiple cloning site with unique restriction site. A short stretch of DNA on vector DNA containing restriction site for possible site for insertion of foreign DNA is desirable. The same thing that we are talking about, suppose there should be site in a plasmid, like we will look into it a little later, I think few slides, where there are multiple cloning sites, basically refers to multiple restriction sites for various different kinds of restriction enzymes, so that you can introduce multiple stretches, multiple types of insert DNA into your vector. Okay. And high copy number. A high copy number is desirable, which can give high amount of DNA after growing host cells, but then there should be certain conditions or criteria where you should be aware of the copy number of the plasmid that is being used, or I mean, yeah, the vector that is being used. So here, suppose in cases where you want to overexpress a certain protein in bacteria, and that suppose that protein can be of some other origin, from insect cells, from suppose uh, from insect cells, it can be from mammalian source, or any suppose yeast source, and you want to express that particular protein in bacteria. So that particular protein might be toxic for the bacterial cell, or because it is a foreign protein altogether, right? And we are like on top of that, we are trying to overexpress that protein. So that particular protein can disturb the bacterial cellular metabolism as well. It can be, you know, like can be a burden for the bacterial metabolic pathways. Okay. So in such cases, if we are using a high copy number vector, which will have a lot of copies or lot of vector DNA that will be there inside the bacterial cell, because it will produce multiple copies of itself in a short period of time. So in such cases, if the protein being overexpressed is toxic to the bacteria or it is disturbing the cellular metabolism of bacteria, then high copy number is not at all desirable. So what will happen is, in such cases, the vectors having low copy numbers should be used, because see, in case of low copy numbers also you will have, like it will be disturbing, it will be toxic, or it can disturb the cellular metabolism again, but then because it has low copy number, the amount is low, so the bacteria can actually potentially uh, can you know, can develop certain pathways or can tide over the toxicity or this particular disturbance that we are talking about. Okay, because it will be slow to function, slow to act upon as compared to a very large amount of toxic substance. And whereas if a small amount of toxic substance or small disturbance is there, that can be taken care of by the bacterial cell. Okay, so bacteria can get adapted to such types of changes. Either it can get adapted or it can, you know, introduce certain changes to it so that that particular small toxicity, or it can either flush out that thing or something, some sort of cellular changes, metabolic changes might happen, that can be taken care of in cases where copy numbers are low. Okay. And now, if we are using vectors just for cloning purposes or for carrying the foreign DNA, then high copy numbers are desirable because definitely high copy number vectors will give you more big amount of DNA. In these cases, suppose where the whatever we are trying to do inside the bacteria, that this won't really harm the bacterial cell, then low copy number vectors, uh, I think this has repeated, then low copy number vectors will give you less amount of DNA and that won't be really fruitful for your downstream applications in case of experiments, laboratory experiments. Okay. So for just for cloning purposes or carrying the foreign DNA, high copy number vectors are desirable. In cases where uh, we are overexpressing certain proteins which can disturb the bacterial cell metabolism, then low copy number vectors are desirable. Okay. Now, there are certain minimum molecular components to assemble a bacterial plasmid to perform the function of a vector. Okay. Now, what things should a particular vector have? Right? It should be small in size. It should have multiple cloning sites that we were uh, seeing. So it should also have an origin of replication, definitely, because otherwise, how can we are talking about copy numbers and all? So if there's no specific site to really replicate its, if to make multiple copies of itself, then uh, it won't really serve the purpose. Okay. So like any other replicating DNA, plasmid DNA needs its own independent origin of replication to provide a start site for replication to make more copies of itself. Okay. It decides the range of bacterial host strain that can be used with a particular plasmid vector. The plasmids containing this origin of replication is also, in short, it is written as ORI. ORI site, ORI site, containing ORI region from col E1. This is just an example. The plasmids containing ORI region from col E1 are able to grow in limited bacterial species like E. coli, etc. And in some cases, E. coli is a gram-negative bacteria. All right. There are certain plasmids that contain the origin of replication site from these strains, RP4 or RSF 1010, that are able to grow in both gram-negative and gram-positive bacteria. We talked about the selective pressure or selective uh, media that we were talking about having ampicillin resistance or some other antibiotic resistance. This is usually the selection marker that we are talking about. Selection marker in the form of either antibiotic resistance gene or enzymatic gene is essential to give phenotypic changes in host after entry of the plasmid. Then there definitely should be a promoter because plasmid replication is host. In host is performed by a variety of host provided proteins, right? Such as DNA gyrase, helicase, DNA polymerase, DNA ligase, and but proteins are also required for conferring antibiotic resistance or enzyme used for selecting transformed host cells is present on plasmid and a promoter adjacent is required to express such genes present on plasmid DNA. All right, suppose we like this, if we want uh, for something like the LAG-Z gene. Okay, we want beta galactosidase activity in a particular cell. Okay, for for some other experiment that we have thought of. So if we want to, so we will be introducing the LAG-Z gene inside the cell that we are talking about, cell of our interest. But then that LAG-Z gene has to express itself also, right? Then only it can really translate and make the galactosidase enzyme. So for that to occur, the promoter for that specific gene or promoter compatible for that for expressing that specific gene should be present. Okay, when we are talking about plasmid DNA. All right. Now, in the bacterial plasmids, plasmids are widely used for cloning of foreign DNA into the bacteria as host string. There can be different forms of plasmids. Okay. Uh, so suppose like we will eventually will be seeing. So when you purify, we saw plasmid purification in the previous week. So what is happening there is uh, after purifying the plasmid, I talked about okay, you should check the absorbance ratios of the obtained plasmid as well as check it running on agarose gel to check how it looks on the gel. Okay. So on gel, Plasmids usually, like the undigested plasmids, usually can take on multiple forms. Okay, there can be different forms of plasmid based on its, like whether it coils upon itself or it has certain links or it is open or linear. Okay, somehow it has been digested. Okay, then it will be linear, completely linear. So there are different forms of plasmids that you get to see on an agarose gel when you run the plasmids directly after purification. Okay, without any further treatment, like you haven't digested it using restriction enzymes or something. Okay, so the different forms of plasmids that can be there. Bacterial plasmid is a double-stranded circular DNA which can primarily or majorly exist in three different forms. If both the strands of circular double strands are intact, then it is called a covalently closed circular plasmid, right? This is the one that we are talking about here. And if one of the strand has a nick, so what was nick that I was talking about? There is an absence of a covalently bond, a covalently linkage. Okay. If one of the strand has nick, then it acquires the conformation of open circle DNA. This one. Like one strand is fine, the other strand has an absence of one phosphodiester linkage is missing. Okay. During the isolation of plasmid DNA from bacteria, covalently closed circular DNA in this one can coil upon itself, thereby forming many number of turns. So what will happen is, like suppose how a rubber band acts sometimes, right? If you open it from your hair or in general, if you open it from any boxes or something, it can coil upon itself. So a relaxed rubber band as compared to the coiled rubber band is more spread out, right? The coiled rubber band is more compact. So this is how a coiled rubber band can look like, right? So this particular configuration or conformation is called supercoiled plasmid. Okay. Now what happens is even even without, I mean, this can be naturally present, but you can also introduce these particular different forms of plasmids using different enzymes. Okay. Suppose this is your covalently closed circular DNA, which is completely relaxed without having any turns upon itself. Okay. Now, if you treat this using suppose endonuclease. So endonuclease will introduce a cut at like cut the DNA at its backbone. Okay. So suppose endonuclease treatment can give us this nick. See this gap is here. So it will result in open circular DNA when endonuclease treatment is given. When the same open circular DNA is treated with ligase, this will join this particular nick, thereby giving us completely closed circular DNA. Right? Now, uh, if we want to introduce these turns into the plasmid circular DNA, there is this enzyme called gyrase, DNA gyrase, that can introduce these loops. Okay, but suppose we have topoisomerase. I think during DNA replication we talked about topoisomerase. Because before replication, the strands are separated, and topoisomerase also comes into play to relax or release the tension on the like the coiling tension on the DNA strands. Okay, so if topoisomerase treatment is given to the supercoiled DNA, what will happen is it can form this relaxed closed circular conformation. Okay, now see, this is the covalently closed circular double-stranded DNA. This is the nicked circular double-stranded DNA. If this what we were looking at in the previous slide. And this is gap circular double-stranded DNA that I was talking about. In case of, we were showing that in case of a linear DNA strand, this is how it looks in the case of a circular DNA strand, like multiple nucleotides are missing or multiple phosphodiester linkages are also absent. Definitely, like nucleotides are missing, so phosphodiester linkage won't be there. But here, only phosphodiester linkage is missing. Okay. Now, PBr322 was one of the first recombinant plasmids to be created. Okay. Now, see here, this has a specific site for EcoR1 restriction. Digestion, this is its origin of replication. And this particular, like the simplest form of recombinant plasmid that was created. and this had tetracycline resistance gene here, represented by TCR, and ampicillin resistant gene here, represented by APR. Now, all these, suppose this particular fragment, tetracycline resistant fragment, was derived from this particular source, whereas ampicillin fragment, resistant fragment was derived from this particular source, and the origin of replication, which will provide the start site for the replication machinery to start its function, was derived from this particular source. Okay. There is another very widely used and very commonly used vector to like in day-to-day laboratories, everyday for everyday purposes. That's PUC19 or PUC19. So PUC19 is the initial example of bacterial plasmid having a small size of around 2.8 kilobases, okay, containing multiple cloning site. So multiple cloning site, what I was talking about is this one, having multiple restriction sites or recognition sites for restriction enzymes, okay. The usual place to keep the MCS or multiple cloning site is always between the initiation codon AUG and the codon 7 in this case that is being talked about. And MCS, like what is the purpose of having multiple cloning site? It allows to design many cloning strategies as as a large number of enzymes or restriction enzymes are available for cloning. Like yeah, in addition to enzymes from this multiple cloning site can be used to insert the foreign DNA without disturbing the plasmid sequences. PUC19 vectors also has a small stretch of DNA which encodes for rapid detection of an insert by blue-white screening. So I was talking, so now I think you guys might know that the beta galactosidase enzyme, if it acts on certain substrate called XGAL, it can produce blue colored products. Okay. So suppose we were talking about selective marker in terms of antibiotic resistance gene only, right? In the presence of antibiotics in this medium to grow the bacterial cells. It can also be by blue-white screening by inserting LAGZ-brown or LAGZ gene construct inside this plasmid, which can code for beta galactosidase. We will look at this in the subsequent slides actually. Okay, which will help us in selecting the marker for, I mean, as a marker for selecting those particular clones having the recombinant vector. Okay, or wherein the cells which have taken up the recombinant vectors. Now, this is one thing just for a basic principle or basic thing. This is the master. Suppose what happens is in PBR 322, we saw there are two resistances, right? Ampicillin resistance gene and tetracycline resistance gene. Now, what happens here is if PBR 322 was to be introduced into E. coli cells, suppose. So then, if the E. coli cells didn't take up PBR 322 plasmid at all, then what will happen is if we try to grow the cells on a medium containing ampicillin or tetracycline antibiotic, none of the plates will show the growth of the E. coli cells, right? But if suppose I have introduced certain gene in PBR 322, PBR 322 has a stretch of ampicillin resistance gene and tetracycline resistance gene. Suppose I have introduced certain gene fragment in the tetracycline resistance gene. So what will happen is the tetracycline resistance property of the PBR 322 plasmid will be hampered. So it won't show tetracycline resistance. So now suppose now I want to select this particular thing, right? So ampicillin resistance gene is intact. Okay. Now if ampicillin resistance gene is intact, then even even if the bacterial colonies are growing on ampicillin resistant gene, I mean ampicillin resistant plate, ampicillin resistant medium, as in medium containing ampicillin, then what will happen is the cells that have only those cells that have taken up that particular PPR322 plasmid, those will be growing on the ampicillin medium, right? But on the tetracycline medium, uh, but actually the cells that have taken up the plasmid can also grow on tetracycline medium, all right? Because like also grow on tetracycline medium. Now, what can happen is if the gene of our interest has been correctly introduced at the tetracycline site, then the tetracycline resistance will be lost, right? So then what will happen is, so the ones inserted, the insertion, if it has taken place correctly, then I would see the growth of those cells only on the ampicillin medium, not on the tetracycline medium. All right. Or suppose a medium contains both tetracycline and ampicillin, so I won't be able to see any growth on such kind of medium. All right. So this is what has been shown here in this case, like uh, when the cells haven't taken up anything. Okay. So then it cannot grow on ampicillin or tetracycline medium. Now, if the cells have taken up the plasmid having both these resistance gene, so only then they will be able to grow on ampicillin or tetracycline containing medium. See, the transformed ones with non-recombinant vector and transformed with recombinant vector. Now, if it has been transformed with non-recombinant vector or the vector has been unaltered, it has taken up the plasmid, but the gene hasn't been inserted at the proper place, suppose. So tetracycline resistance is intact. So what will happen is it can grow in both ampicillin and tetracycline containing medium. But if the gene has been inserted at the suppose tetracycline medium or ampicillin resistance gene part, then if the like the gene of our interest, the transformed recombinants can grow only in ampicillin medium and cannot grow in tetracycline medium due to insertional inactivation, because the tetracycline resistance gene has been disrupted by the insertion of another gene, which is basically the gene of our interest. Okay. So recombinant colonies can be easily selected from this master plate. Okay. So I think you guys remember when colony hybridization, we were reading about colony hybridization, etc. They were creating replica of the bacterial plates, right? So this is how that can be created, and the same colonies can be, grown and seen if which type of colony is growing in which type of medium, like containing which type of antibiotic. Then we can actually select out the exactly those particular colonies from that master plate which has the which has our colony of interest, which is the transformed ones with recombinant vector, which is having the gene of our interest inserted at exactly the specific site that we wanted to. Okay. I think plasmid DNA extraction, we looked at it the previous week. Let's go through it very briefly. Like you will be lysing the cells, definitely pelleting the cells down, culturing the cells, pelleting the cells down, lysing it out. And by lysing, you are actually uh, breaking the cells open to take out chromosomal DNA, plasmid DNA, proteins, and other components of the cell. You centrifuge it again using different kinds of solvents. Then either you use chloroform phenol extraction that we saw in the previous week lectures tutorial, or you can use spin column, etc. And then wherein you selectively let the plasmids stay in a particular phase of a solution, or selectively let the plasmid selectively bind to the spin column filters that we are talking about, silica filters. Okay. And then precipitate out the DNA, plasmid DNA using alcohol. Okay. Wherein alcohol is disrupting, basically precipitating out the DNA by disrupting its interaction with the solvent. Okay. Or its interaction with the silica also. All right. So just quickly we will look at it because this is the same slide from the previous tutorial also. So plasmid DNA extraction is trickier because the plasmid DNA must be kept separate from genomic DNA to avoid cross contamination. So lysis is usually done with sodium hydroxide and detergent SDS, which completely denature your plasmid and genomic DNA, basically separating DNA into single strands. This is crucial because excessive denaturing can result in an irreversibly denatured plasmid. Then you actually neutralize it using potassium acetate so that the renaturation can be done, and this should be quick, wherein the genomic DNA having much larger size takes a like it basically becomes entangled and takes a long time to renature, but plasmid DNA being smaller, it renatures quickly. Okay, plasmids are small, they can renature or reanneal quickly, but genomic DNA can take too long. And then by centrifugating the whole sample, genomic DNA which is bound to proteins can form a pellet and separate out from the plasmid DNA, which remains soluble. Okay, and then you finally purify it. Plasmid DNA is in the supernatant. This can be precipitated with ethanol that I was talking about. Ethanol disrupts its interaction with the solvent, or it can also be cleaned up using phenol chloroform, wherein the plasmid DNA is present in the aqueous part, right? Or a spin filter, wherein the plasmid DNA selectively binds with the silica present in the spin column filters. Okay. So this is what is written here in spin filter technology. Neutralization buffer will contain guanidine salts so that the lysate can bind the silica directly for further washing and elution, and the resulting DNA is pure enough for most of the downstream applications. Okay. Now, this was what I was talking about. These are the like after purifying your plasmid, these are the different forms that you get when you run on agarose gel. Nicked, linear, supercoiled, and circular single strand. Supercoiled are the ones that have multiple turns. Linear is the one that has been digested or cut at both the double strand, like both the strands, and nicked is the one only one, like at only one strand or one strand. There's a missing phosphodiester linkage. Okay. Now let's go through it one by one. Supercoiled DNA is the native DNA conformation found in vivo, which means inside the cells, and occurs when extra twists are introduced into the double helical strand. Alright? In the case of DNA plasmid preps, this superhelical tension cannot be relieved because the end of the plasmids are joined together. Right? Supercoiled DNA migrates faster because it's more compact and its hydrodynamic radius that we are talking about, the overall size, reduces, right? So it can very easily pass through the pores, whereas something as big as this and something like this, something which is more spread out, more broad, have like something which has a larger hydrodynamic radius can find it like can encounter more obstructions during its passage through the agarose gel pores. Alright, alright. But something which is more compact can easily pass through the pores. So in case of joined together, supercoiled DNA migrates faster than predicted in an agarose gel due to its conformation. Okay. Supercoiled DNA is the desired species when isolating plasmid DNA, which suggests that your DNA is completely intact, right? Without any nicks or without any other single-stranded portions present in your DNA. Okay, present in your plasmid. Next comes DNA found in the supercoiled form is not easily accessed by the replication machinery. So, during replication, cellular topoisomerases can nick one strand of the DNA helix and relax the superhelical tension, thereby allowing the polymerases to gain access to the DNA. Right? So, this is the nicked form, which actually runs, which is the largest of all due to its overall size, which runs the slowest on an agarose gel. All right. So why these different forms? Why do we have cellular topoisomerases in this particular context? Because we are actually isolating plasmids from cells that have been cultured, and then we are pelleting down those cells. So those cells have their metabolic activities actively going on, right? So some plasmids might be at this particular phase where replication was about to begin, suppose. Okay. So it might have this nicked strands, or most of them should be like are in their supercoiled form. Some might be linear, like both the strands have been cut. Okay, so this is because the cells have been harvested at the time when their metabolic activities were going on naturally, right? Now linearized DNA, this one, occurs when the DNA helix is cut at both the strands at the same place, right? Now, linear DNA generally migrates between this knit circle and the supercoiled forms. However, it may also migrate the same distance as the knit circle sometimes. Okay. Now, if you get linear DNA, when you are actually, suppose when we actually do plasmid preps, we actually hope for this particular species. And this should be, this is you see, this is a thicker band. This ideally should be more in number. If you get linear like linear DNA, when you are hoping for supercoiled, for example, DNA after plasmid preps, it is due to nucleus contamination or harsh treatment during purification. Okay, this can also show or give you an give you a quality check kind of to show that okay how your plasmid prep was, how good your plasmid prep was. Okay. Now alkaline lysis plasmid preps disrupt hydrogen bonds by the alkaline conditions, sodium hydroxide that we were talking about. Now if in cases where alkaline lysis step is too harsh, see in that step we talked about denaturing the plasmids, both plasmid DNA as well as genomic DNA using sodium hydroxide, right? Yeah, sodium hydroxide and detergent. And then you neutralize the solution quickly so that the plasmids can renature, but the genomic DNA, which is due to its size, takes time to reanneal or renature, right? So in cases, suppose if it's for too long, this particular step, the alkaline lysis step is carried out for too long, incubated for too long, then DNA can permanently denature, giving single-stranded, like the reannealing won't be happening, thereby giving single-stranded closed circles that definitely will migrate much ahead of all the other forms. Okay. But again, the circular single-stranded ones will mostly be useless in most of our experiments. We want ideally the supercoiled form. Then the further processing can be done to create linearized DNA or linearized plasmid DNA as well for some other applications or other experiments. Okay. Now, alpha complementation process, we were talking about blue white screening. So what happens here is when lactose in the surrounding medium is present, it triggers the Lacz operon in E. coli that produces the beta galactosidase enzyme, which metabolizes the lactose. Okay, and this beta galactosidase enzyme actually exists in a tetrameric state, which is a homotetramer. Suppose alpha alpha alpha, four alpha subunits, kind of. Okay, in its active state, suppose hemoglobin has alpha and beta subunits, so that is a heterotetramer. And in case of beta galactosidase, it is formed of four similar subunits. Okay. Now what happens is there's a certain kind of mutation, M15 deletion mutation. What happens is the N-terminal part, beta galactosidase is a protein, right? Enzyme, this enzyme, the N-terminal part of the enzyme, few residues are deleted. So that deleted and the particular peptide that is being formed with the deleted N-terminal residues is called omega peptide. Okay. Now this omega peptide with its deleted N-terminal region is unable to form a tetramer and is inactive. Okay. And the fully active tetrameric state is only possible in the presence of the N-terminal residue. Okay. Now what happens is the N-terminal residues are deleted. The remaining sequence that is formed, the peptide that is called omega peptide. And the deleted N-terminal residue can also form a particular peptide. Okay, that is called the alpha peptide. So neither the alpha peptide or the omega peptide is functional by itself. Only when the alpha peptide and omega peptide come together, then only a fully functional beta galactosidase activity can be there. Okay. A plasmid containing LAC-Z alpha sequence, which codes for the alpha peptide, are transformed into M15 cells, which actually contain the deletion at the N-terminal region. Thereby, these cells code for omega peptide and some other plasmid, which is which only codes for the first few residues which have been deleted from the N-terminal region, which codes for the alpha peptide. That plasmid is put inside these M15 cells. So the two peptides, both the alpha peptide from the plasmid and the omega peptide, which was already present by the cells, are expressed together, thereby forming a functional beta galactosidase. So the rescue function of this mutant beta galactosidase present in these M15 cells by the alpha peptide present in the plasmid is called alpha complementation. Okay. Now, how is this alpha complementation process being used? Okay. So this is used as a marker for recombination. Okay. What happens is a multiple cloning site is present within the LAGZ sequence in the plasmid vector. Now, this sequence can be nicked by restriction enzymes to insert the foreign DNA. Okay. When a plasmid vector containing foreign DNA is taken up by the host E. coli, the alpha complementation does not occur. Therefore, a functional beta galactosidase enzyme is not produced. Okay. If the foreign DNA is not inserted into the vector, or if it is inserted at a location other than the cloning site, the LAGZ gene in the plasmid vector can complement the LAGZ deletion mutation in the host E. coli, producing a functional enzyme. So, what did I mean by this? Let's look at what is happening here is, suppose I referred to X-gal, right? This particular substrate. So beta galactosidase we can screen the colonies with recombinant DNA. Suppose X-gal is a chromogenic or color producing substrate which is added to the agar plate. Now, if a fully functional beta galactosidase enzyme is produced, it will hydrolyze this X-gal substrate and form subsequently an insoluble blue pigment. Okay. So colonies that are formed by non-recombinant cells that don't have an insert, which hasn't hampered the property of the like hampered the formation of beta galactosidase. So those will have normal beta galactosidase activity, thereby appearing blue in color because the X gal is present in the medium and that will actually um, hydrolyze this substrate to give blue colored product, while the recombinant ones which will produce non-functional beta galactosidase, which will hamper the coding of like hamper the functioning of fully functional or fully active beta galactosidase. Those recombinant colonies or recombinant cells will appear white definitely because those won't be able to hydrolyze this particular color producing substrate. So exactly by looking at the cells, we can pick out the colonies having our recombinant DNA and non-recombinant DNA. Like we can distinguish between the recombinant cells and the non-recombinant cells. Now, in order to have the beta galactosidase produced in large amounts, I think we discussed about IPTG, which induces the expression of LAGZ. And we should also be careful that IPTG is not the substrate for beta galactosidase. In this scenario, XGAL is the substrate here, but it induces the activity of beta galactosidase. For visual screening purposes, chromogenic substrate like XGAL is required, and IPTG is just for inducing the expression of the LAGZ gene. So this is what I was talking about. Suppose this whole medium that you see, this yellowish part, okay, this is the whole food for the bacterial cells. Now this has XGAL substrate, that color producing substrate we were talking about. Now the cells that have taken up the plasmid or the insert has been there. So they will produce non-functional beta galactosidase enzyme. So that non-functional beta galactosidase won't be able to act upon the X-gal present in the medium, thereby producing white colonies. And where the insert has actually taken place, which so these are the ones where the insert has actually taken place, suppose. And in cases where beta galactosidase enzyme activity is completely intact, this will give out this blue colored colonies, okay, because this substrate is like already present in the medium. So this will be acted upon by the beta galactosidase enzyme and this will produce blue colonies. So these blue ones suggest that the sequence of our interest has not really been inserted because the beta galactosidase activity is intact. These white ones suggest that these cells have actually taken up the insert and the insert has been the sequence of our interest has been inserted at the correct position because it has hampered the activity of beta galactosidase enzyme. All right. Now this is what I was talking about. Blue-white screening. This is plasmid vector. This is foreign DNA. Now this is the LAGZ gene. okay, present in the plasmid vector. Now this is the red one is the foreign DNA or insert DNA we are talking about. If the insert is within the LAGZ gene, so the LAGZ gene won't be coding functional beta galactosides enzyme, right? So in the presence of XGAL and IPTG, white colonies are there, what we saw in the previous one, previous picture. If the insert is, suppose it has inserted, but um, definitely like the insert is there, but it hasn't really inserted at the specific position, then we will get functional beta galactosidase enzyme producing blue colonies. And if again the cells haven't been inserted at all, then also we will get blue colonies, right? Okay. Now let's move on to the questions one by one. Uh, so think okay, we have 10 minutes left. We will try to finish this up. Now, the removal of the terminal phosphate groups from DNA is done by, like we just went through it in the lecture. So can anyone answer? Yeah. C, right? This is alkaline phosphatase. Okay. This will remove the terminal phosphate groups. Next, the EcoR1 methylase adds a methyl group to which residue in the EcoR1 restriction site? Does anyone remember? Yeah. See here, this is the same picture from the previous slide. This is the A residue that is being methylated. Okay, acted upon by EcoR1 methylase. Next, so correct option is A. The cleavage site of type 1 restriction enzyme is how many base pairs away from the restriction site or recognition site? Can anyone answer? 1000, yeah. Option A. Okay, correct option is A. Now, which of the following subunits of type 1 restriction enzymes provides specificity for the recognition site? Like here we are talking about specificity. A is HSDR. It will be S. Okay, R is the like restriction activity or the exact cutting activity, like endonuclease activity we are talking about. Okay, S is for specificity, recognizing that specific sequence, and M is for modification or methylation activity, right? So correct option is HSDS, option C. Now the restriction enzyme that cuts methylated DNA is, can anyone take a, can anyone try it out? So the option is D, because all these ones, EcoR1, see we talked about EcoR1, right? It has R, M, S, all these subunits, right? All these activities present. So if it is methylated, this won't be able to perform its function. Won't be showing the restriction activity. So this is a DPN1 enzyme which can actually cut only recognize methylated DNA strands and degrade it. Okay. So the option, correct option is D, DPN1. Okay. Now the BAM-H1 restriction site is, does anyone know the answer? Yeah. It is GGA-TCC. And what about this one? GAA-TTC? Yes. 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 Okay. Now, the molar ratio of vector to insert for a ligation reaction is ideally, um, I think from the lecture videos, the correct answer will be 1 is to 3. But actually, you know what happens in case of ligation reactions, uh, people can go up to 1 is to 5 ratio, 1 is to 7 ratios as well. Okay. So for typical cloning applications, uh, Again, I was talking about enzymes for setting up any ligation reaction or restriction enzyme reactions. What will happen is for, for, for cloning things, the company from where you are getting your specified reagents, getting your required reagents, the reaction conditions can also depend on that company specified things, but it is more or less the principle is same. So suppose 50 nanogram of vector is recommended, which will approximately be this much, these many moles, depending on the vector size. Now for inserts, having sizes similar to the vector, 1 is to 1 molar ratio is recommended, right? Now, if the insert size is much smaller than the vector, what will happen is, suppose 1 kb insert has to be ligated into 3 kb vector or 5 kb vector, all right? Or 500 base pair insert has to be ligated into a 3 kb vector. Then what will happen is you need a much higher amount of insert, that is the smaller fragment, so that the vector molecules, which is much larger, they have higher chance of integrating into the vector sequence. All right. If the insert is much smaller than the vector. Now, in cases where the inserts are really small, like maybe 100 bases or something, or in the range of 2 digits, then even higher ratios can also be used, like 20 is to 1. So here, when we talk about 20 is to 1, 20 is the insert ratio, 1 is the 20 is the insert amount, 1 is the vector amount. Okay. So amount means suppose these many number of moles are being used. This is the molar ratio. Okay. So if these many moles are being used, these many moles of the vector is being used. So okay. Suppose or millimoles of vector is being used. So basically this is the vector insert in is to vector ratio that is being used. Okay. Now there are multiple online calculators. Here in this case there is NAB calculator where you can directly put your ratios and you can calculate. I can just give you an example here. Uh, See, your insert DNA length you should be knowing. Okay, it can be in base pairs or suppose it's 100 and your vector DNA length is suppose 5 kb or 3 kb something, suppose 5 kb. And you have to give your vector DNA mass definitely, and usually we keep it at suppose 100 nanogram or 1 microgram, whatever is the thing. Okay. Suppose if this is the thing, see automatically you get this particular thing. In case where you are using 1 is to 1 vector is to insert ratio, then you should be using insert DNA of 2 nanogram. Now, how do you get these masses? Okay, so when you actually calculate the concentration of your uh, from the molar ratio only, you can get this particular value. Okay, or so this is how you can calculate directly. These are the online calculators. These are some sites. This is NEB calculator where you can get this particular kind of different kinds of ratios which you can try out. See, I was talking about 1 is to 7, 1 is to 5, 1 is to 3. So different kinds of ratios can be tried out. Okay. Now, sorry. Now, which of the following sites for origin of replication allows the vector to replicate in both gram-negative and gram-positive bacteria? See, the answer is here in this paragraph only. So the answer will be option. Sorry, can you repeat? Um, can you repeat your thing because there was some disturbance? Yeah, yeah, it will be A. Okay. Yeah, yeah. Okay. So option is A. Now, we were talking about the different forms of plasmid DNA. A covalently closed circular plasmid, when exposed to which enzyme, can transform it into an open circular form. Covalently closed means the relaxed form that we are talking about. Open one is the nicked one. All right. So what will be the answer? Can you repeat it? It will be nuclease enzyme because it will be a nuclease. Okay, because it is creating a nick. When it is an open circular form, it is having a nick there, right? Now, now, okay. If it is a nick, so it can be a nuclease. Now, what should be the one? Whether it's endonuclease or exonuclease? Exonucleases are the one that cuts at the terminal part of the DNA sequence, and endonucleases are one which cuts at the internal sequence, okay, which cuts in between the DNA nucleotides. Now, in case of a plasmid DNA, this is like entirely, there's no terminal point, all right. So this will be endonuclease, okay, option B. This will definitely be a nuclease because that is creating a nick, thereby creating an open circular form. But what kind of an what kind of a nucleus? This will be an endonuclease. All right. Suppose in case of a linear strand, we could have thought about okay whether it's an exonuclease or endonuclease or something. But here we are talking about circular forms of DNA. So their exonuclease won't be there in the picture. Okay. Now the ampicillin resistant gene of PBR322 is derived from. See, the answer is in the picture only. This is the ampicillin resistant gene. So the answer will be option this one. Yeah, RSF2124. Okay. This is the ampicillin resistance gene that has been derived from this source. This is the tetracycline resistance gene that has been derived from this source. Okay. Now, we just read about blue-white screening. So, blue-white screening cannot be performed with bacterial colonies transformed with which of the following vectors? Can anyone give it a try? I think the answer is also in the slide itself. So, for blue-white screening, what do we need? We need the activity of beta galactosidase, all right, to check its activity on the XGAL product. Now, for beta galactosidase activity, which particular gene do we need? We need LAGZ gene, right? So, in the PUC19 case, in the PUC19 vector that we saw, PUC19, yeah, see, there is this LAGZ gene here, right? But in case of PBR322 vector here, do we see any LAGZ gene insert here? No, right? So, blue-white screening cannot be performed where the LAGZ gene insert is not there. So, in all the other plasmids, PET28A, PET26B, PUC19, LAGZ gene insert is there, which then we can use blue-white screening uh, to check the recombinant and non-recombinant cells. Okay, but PBR322 we can't really use blue-white screening. Okay, so the correct option will be option A. All right. Next, I think most of you will be able to answer the question. What is the correct sequence of steps during plasmid isolation? Can anyone try? Option B. Yes. Alkaline lysis, renaturation by neutralization, deproteinization, alcohol precipitation. Okay. Option B. Okay. Now, so we were talking about different kinds of antibiotic resistance. So the tetracycline resistance gene produces which kind of, whether it's a molecule, whether it's a metabolite or protein, what happens? Like for this, you guys should be knowing about what do each, like what does each type of antibiotic do? Okay. So what happens is in case of tetracycline, it actually hampers with translation activity of the cells. Okay, it binds at the ribosomal sites A, P, E sites. There are three sites, right, during protein synthesis. So it binds at one of the sites and hampers protein synthesis. So if something is resistant to tetracycline, what might be the case? What will happen is ribosomal protection proteins will be formed. Okay. If something is resistant to tetracycline, then ribosomal protection proteins will be formed, which will actually counter the effect of tetracycline. All right. It will not help the tetracycline to really go and bind to the ribosomes. So what happens here is tetracycline resistance is associated with TET genes. These are the genes, like these genes are abbreviated as TET genes, which are involved in active efflux of the antibiotic. Definitely, they will let the antibiotic bind at its specific site. They will these particular genes will code for certain metabolites, which will help in removing the antibiotic, and it will definitely protect the ribosome, thereby inhibiting or hindering the binding of tetracycline drug and enzymatic degradation of the antibiotic. Okay, so these genes code for all this. So the correct answer will definitely be option A, like the genes which are resistant to tetracycline, these code for ribosomal protection proteins. Now, beta lactamase, this is produced by ampicillin resistance gene, and all these are involving other, like involving other antibiotic resistance cases. Okay. So, but in case of tetracycline, this is option A, ribosomal protection proteins. Okay. Now, I think this is also from the lecture videos. Bacteriophage lambda-based vectors. What happens is bacteriophage cloning vector has a middle segment responsible for insertion or excision, and this region can actually be replaced by any foreign DNA of our interest. Okay, with the help of two restriction sites present on either side of this insertion or excision region. And what happens here is this green segment, which is this segment after now this particular segment has sites which can produce overhangs corresponding to BAMH1 digestion. And our DNA of interest can also be digested using BAMH1 so that the uh, compatible sticky ends are produced. Thereby, now if this particular gene of interest is uh, incubated with this and using DNA ligase, then what will happen is this can get inserted and inserted into this bacteriophage lambda. This is actually a virus that infects E. coli cells. All right. So in general, what happens here is these are the different kinds of vectors that are there, like EMBL4 vector, EMBL3 vector, wherein there is this left arm, there is this right arm, there is this central region or central stuffer region having restriction sites at both the ends. So when we usually want to really put in our DNA of interest or any foreign DNA, what people usually do is chop off this particular central stuffer area like that. See, these are the specific restriction sites present here. Then by creating like compatible ends, and the foreign DNA can be inserted in this area. Okay. So this is basically the concept, because based on this, the last two questions are present. What happens here is also this particular, these ends are, in case of bacteriophage lambda DNA, these cohesive ends are the cos ends. Okay. Now the cos sites are present in, like, a very straightforward answer, bacteriophage lambda based vectors. Okay. Correct option is D. Now, which region of the EMBL3 vector is replaced by the insert? We just now looked at it. Central stuffer region, because it has restriction sites for like recognition sites for restriction enzymes flanking both its ends, so that this can be chopped off and a new DNA can be inserted. Alright. So the correct option will be option E, central stuffer region. Which region of the EMBL3 vector? This is central stuffer region. Okay. So that was for today's class. Let's stop my presentation. Okay. Okay then. See you next week again. Thank you for joining. Yeah, thank you so much. Okay.